Greetings, listeners, and welcome to this episode of Cell and Gene, the podcast. Thanks for tuning in. I'm your host, Erin Harris, and I'm joined by Dr. Paul Lammers, CEO of Triumvira Immunologics, a clinical stage company developing novel, targeted autologous and allogeneic T-cell therapeutics that co-opt the natural biology of T-cells to treat patients with solid tumors. And we're going to break all that down in just a minute. But first, Paul, welcome to Cell and Gene, the podcast. Thanks for being here.
Thanks, Erin. Great. Thanks for inviting me.
Absolutely. All right. So, of course, I did some research on Triumvira Immunologics before our discussion today, and your technology attempts to overcome the barriers of current T-cell therapies by harnessing the power of the natural TCR. So, but before we get into all of that, I would love to hear from you your take on the current barriers of T-cell therapies where we are today.
Okay. Well, let's start off with the biggest group, which of course are CAR-Ts. We all know chimeric antigen receptor technologies that have, quite a few have been approved now for liquid tumors. So lymphoma, leukemias, multiple myeloma, but not for solid tumors yet. Solid tumors provide a separate challenge. But let's talk about CAR-T. So CAR-T is a beautiful, simple design. It is a small structure that is expressed on T cells and then infused, of course, in the patient. The CAR-T structure works independently of the normal T cell receptor in a cell. It has its own activation domain. It has its own co-stimulatory domain. So CAR-Ts are built to produce cytokines. So when you manufacture CAR-T cells, even during the manufacturing process, they start producing cytokines because that's how they're built. So these cells, poor cells, have to run a marathon, which means and also they provide so much cytokine, so many, that they leads to well-known toxicities like cytokine release syndrome, to ICANS, neurotoxicity, but also they run out of steam because again they have been overproducing cytokines. So that is the biggest challenge right now for the CAR-T space. It's what you call premature exhaustion, so they tire early, and lack of persistence. Right now, in the heme setting is relatively easy because these tumor cells are either in your vascular system or in your lymphatic system. So it's easy for the CAR-T cells that you inject into patients to find the tumor cells. Solid tumors is a different ballgame, and we'll get back to that later. So I think that is the biggest group. Now, people have tried to use different type of cells, whether it's natural killer cells, NK cells. Now people are using iPSCs or pluripotent stem cells to try to have the own patient's immune system evolve and produce the kind of subcells that they need to basically attack the patient's own tumor. And I think that is a key issue because we also working in a backdrop of the host immune cell, right? The patient's own immune system, right? So the immune system has evolved over millions of years in humans and is extremely smart in identifying challenges, whether that is invasions like bacteria, viruses, parasites, you name it, right? So, but also therefore, if you bring something new into a patient, even like an engineered T cell, although the T cell may come from the patient itself, the way it's engineered, the immune system is very smart. The human body is very smart. So that is the biggest challenge. Do you get issues like the toxicity that I mentioned? Does the body react to that new, a mechanism that you bring in? And that, for instance, still is when you can, for instance, gene editing is a big thing right now with CRISPR and Cas9, the whole thing, which is a beautiful system for gene therapy issues. But we need to be careful because the body might sense that there's something different about these cells that you introduced, right? So I think that's another challenge in the T cell therapy space. And then I think that, as I said, solid tumors are separate because if you inject cells into a bloodstream in a patient that has a solid tumor, whether it be lung, breast, pancreatic, you name it, that cell then has to extravasate out of the bloodstream, go into a stroma, find that tumor bed, get into the tumor cell, and then basically attack the tumor cell. So That stroma that I mentioned is often referred to as the tumor microenvironment, which is in cases considered a hostile because there's a lot of local factors that also protect the tumor. In fact, the tumors are really smart, so they affect the local immune system around the tumor cells and the tumors in these organs. So that hostile tumor microenvironment is another reason why T cell therapy sometimes or cell therapies may not be effective in the solid tumor setting.
Sure. Okay. Thank you for breaking all that down for us. I appreciate it. I want to talk a little bit about TAC,
Yep.
which for our listeners is Triumvira's proprietary T cell antigen coupler,
Correct.
TAC. And it has both autologous and allogeneic properties. So I'd like for you to talk to us about TAC T cell therapy. What is it? How does it work? And I'd like to start with auto, and then perhaps we can move on to allo.
Sure. Well, let's go back to the starting of the company. So Jonathan Bramson and Christopher Helson, two scientists at McMaster University in Hamilton, Ontario, Canada, started to work on CAR-Ts. And they said, look, the CAR-Ts were effective, but we killed so many animals in the lab, it was ridiculous. So they said there must be a more natural way of activating T cells. So they started to tinker and try to come up with different vectors instead of a CAR that could activate the T cell, but without the associated toxicity. So they tested a lot of different ones and they ended up on the TAC, or T cell antigen coupler. It's also a chimeric antigen receptor, just like a CAR, but the structure and biology are completely different. So as an example, the TAC structure does not have its own activation domain. It doesn't have a co-stimulatory domain. All the activity of the TAC goes to the normal T cell receptor. And that is really critical because leading scientists in the space like Michael Sadley and Memorial Sloan Kettering of Crystal Makel at Stanford also say, look, using the normal TCR is important. Why? You need to activate the cell to the normal T cell receptor, but also silence the cell when the cell does not need to be activated. And that is really, really important, especially in the solid tumor setting. So I think that the benefit of the TAC, so the TAC has three domains. And on the top, it has a binding domain that binds to an antigen presented on the cancer cell, just like what you put on the top of a car. It's just a tumor-associated antigen binding domain. Then it has a domain that binds to the outside of the normal T cell receptor, the epsilon domain of the TCR. So all the activity therefore goes to the normal TCR. And thirdly, and we're unique in the space, we have part of the CD4 co-receptor, also part of the vector, that has two components. It has basically a membrane binding domain that anchors the TAC in the surface of the T cell, but it brings in an intercellular domain that contains in the kinase domain known as LCK kinase domain, that activates the intracellular activation motifs or items of the normal T cell receptor. So that is really responsible for activating the T cell or silencing the T cell when the T cell does not need to be activated. And that is really, really important. You need to give the cells rest as well, because a T cell has a certain life, right? As I mentioned, the CAR T cells, they basically exhaust prematurely. The TAC T cells don't have that because they get into a rest phase, and also therefore they show persistence. So the biology and the structure of the TAC is completely different than a CAR. Because of that, also we have received very strong, compositional matter patents for the TAC technology, and it really has a more natural way of activating the T cell and silencing the T cell when it does not need to be activated.
All right, thanks, Paul. So I'd like to talk a little bit about your pipeline, or at least a portion of your pipeline. So we have TAC 100 HER2, which is under development for targeting relapsed or refractory HER2 positive solid tumors. And we also have TAC 101 Claudin 18.2, which is under development for targeting relapsed or refractory Claudin 18.2 positive solid tumors. Talk to us a little bit about each one, where we're at, and what we can expect going forward in the near term here.
So I would say about 5 years ago, our research group started the whole process of bioinformatics analysis, looking at ideal targets on solid tumors. They identified, they looked at 69 different targets and they identified the top 10. HER2 was number one, and then we have Claudin 18.2, Gucci2C, and GPC3 as the following basically pipeline products, right? Now, HER2 is very well known. It is expressed on a lot of solid tumors. Everybody knows Herceptin in breast cancer as the most prevalent and prominent product here. The challenge with, so HER2 is expressed on about 15 different tumors. The challenge is in this space that a lot of tumors ultimately develop resistance against their therapies. So there's always the need for new targeted therapies. So based on all the preclinical work that we did on HER2 targeting with our tag, we found that we saw, immensely, supportive and great in vitro and in vivo data. So that's decided to bring that first into the clinic, targeting solid tumors that overexpress HER2. We have completed our phase 1. It's been extremely safe. We see some interesting responses in gastric and gastroesophageal cancer. Cancers with high unmet need. Gastric cancer is about the 5th most prevalent cancer in the world, and it's the number 3 leading cause of cancer death. And so therefore now we have started our phase 2 targeting purely gastric and gastroesophageal cancer, because that's what we think is low-hanging fruit for us to continue and try to move that towards ultimately commercialization. Claudin 18.2 is behind it as our second. Both of these programs are autologous, and I'll go into that in a minute. So that is targeting also gastric cancer, but also pancreatic. We're also looking perhaps at non-small cell lung cancer and ovarian cancer. Claudin 18.2 has attracted a lot of interest in the last 2-3 years. A lot of companies now pursuing it. 3 years ago, nobody knew about Claudin 18.2. Suddenly it's everybody's favorite target. And the nice thing about Claudin 18.2, the differentiation, for instance, Claudin 18.2 versus HER2, HER2 is expressed not only in tumors, but also in a lot of normal cells, for instance, in lung and heart. So if you think about Herceptin, Lapatinib, and HER2, some of these products that are targeting HER2, they are known to have what you know on-target off-tumor side effects. So, pneumonitis, so problems in the lung, endocarditis. I mean, all these things are side effects known to immuno-oncology drugs that are targeting HER2. Claudin is only purely expressed in cells of, the gastrum, your stomach, or your pancreas, which means there's no risk or huge limited risk of an on-target off-tumor effect, because it's really restricted to tumor cells only, which is ideal. And then we have behind this, we have Gucci2C, which is a great target for colorectal cancer. And GPC3 is pursued by different companies and also by us ultimately in the future for liver cancer, primary liver cancer. So that's how we build our pipeline, starting with the low-hanging fruit with HER2 now, focusing on gastric, Claudin also gastric, pancreatic, and then moving on to other targets in the future.
That's fantastic. Next year, perhaps this time we can come back and have an update on where we are with those trials.
So perhaps I can answer your question about autologous versus allogeneic. So basically, we are using autologous, i.e. patient-derived cells, right? We take blood from a patient, we isolate the T lymphocytes, we send it to the lab, we add the tag to it, we expand it till we have billions of cells, we send it back to the clinic, and the patient gets his own cells back. But now it's armed with that kind of, whether it's a CAR or a tag on it. So that's how the autologous world works. Autologous manufacturing is cumbersome. However, we are the first company in the US to use the cocoon from Lonza to manufacture autologous T cells. And it has been an absolute game changer in the world of autologous manufacturing. We have 100% manufacturing success rate. It is an incredible machine. And so the last few years, Erin, when I talk to investors or pharma companies, they say, Paul, we want allo, or allo. And now they say, by the way, you have this amazing machine, and autologous is really a huge benefit. Now, also because allogeneic, or off the shelf, I should call it, has gotten into trouble a bit. And the reason is the lack of persistence. So again, in lymphoma, for instance, if you take an allogeneic CD19 product, which of course CD19 is the target in lymphomas and leukemias, what you see is that it works well, but after a month, the cells are exhausted, they don't work anymore. An autologous CD19 works 5 months. So the hematologist said, guess what my favorite is? I want rather give something that works for 5 months than something that works for 1 month, right? Now, there's benefits to using allo, because you can create an off-the-shelf product and literally take it off the shelf and prepare it for a patient, which means is you can administer this product to a patient in about 5 days. Autologous takes about 3 weeks. So that's a benefit, especially patients who have a very aggressive tumor. It's great to be able to, to treat that patient as soon as possible, right? So also you create these master cell banks, right? Which means is you can create with a single, product that you create, you can do 200 or 250 doses, which means is, it is much cheaper to manufacture as well, allogeneic. Sure. So there's clearly benefits to it, but the challenges are that there's lack of persistence right now. So for allogeneic focused companies, they're trying to come up with ways that it works longer, right? So I think that's the challenge. So for us, we think it's very important that we have 2 manufacturing platforms in the company. We have an autologous using the cocoon from Lonza. Mm-hmm. We have an allogeneic. We're using gamma delta cells, which is a subset of the T cells that have an ideal profile for developing an allogeneic off-the-shelf system. Why? Gamma delta T cells, as I said, are a subset. They live in peripheral tissues already. So they also do normal immune surveillance of tumors. And in fact, they have anti-tumor activity themselves. So by adding the tag, we might get a double whammy here, to be able to find the cells in the organs, in the tissues, and then get into the tumor. So for us, that is something that we're looking, forward to bring it to the clinic soon, perhaps as a follow-on product. So our advisor said, look, bring your products into the clinic as an autologous, and then have an allogeneic version as a fast follower. Do it a year later or so. So that's something that we're working on as well.
Yeah, that's fascinating. And that actually kind of leads me into my next question quite seamlessly. And in that, I wanted to talk a little bit about what the next few years of cancer treatment evolution really looks like. And is it the marrying of the two, like what we're trying to do here? If you could, for us, paint a realistic picture of allo and auto and their potential.
Yeah, sure. Well, look, first off, everybody's always hoping to find the cure for cancer, but we need to be realistic, right? I think that finding a cure, yes, there are certain cancer types that we've been able to very, very effectively treat, right? Like take testicular cancer as an example, but there are so many tumors that are really, really difficult. And as I said, tumors are smart, right? I mean, they find ways around blocks that you put into place. And so therefore, if we can turn cancer into a more well-controlled, longer-term disease, but with a really good quality of life for the patient, that would be ideal. And I think that's right now what we're trying for. It's very important that we think about combination therapy, right? So, and also the quality of life is important, to mention that more than 50% of all cancer patients in the world, Erin, are still treated with platinum-based chemotherapy, which we know is horrendous from a toxicity perspective, but it's sad. So it really destroys the quality of life in these patients, or it's just miserable to get those kind of therapies. So if we can get to combination therapies that have a much improved safety profile, but work well together. For instance, now you can say, I'm going to combine an ADC, an antibody drug conjugate product, with a small molecule, or I do a cell therapy approach, together with a monoclonal antibody approach. I mean, all these combinations is what really is going to drive the future of cancer therapies. And then, as I mentioned earlier, now, the gene editing and using messenger RNA combinations with a CRISPR approach, all these things will evolve and will take up a bigger, not only attention span, but also more funding. And, it's amazing the science and the creativity of the scientists in this space. So I'm really excited about what's going to happen in the next 8, 10, 15, 20 years, for cancer treatment.
Good. That makes me feel good, and certainly our listeners too.
Yeah.
Okay, we've reached kind of the formal end of our episode. And at the end, I like to talk to my guests to see who they are when they're not developing life-saving therapies. And so for you, I wanted to know if you had to create a 2024 bucket list, what one or two things would be on that short list, would you say?
So my wife and I love to camp, so we bought ourselves a roof nest. You may know, one of these things that you put on top of your SUV, you can unfold it. So we're very excited about using that this year. And we love to. We did, of course, we're Dutch originally, although I'm American now, but so we saw a lot of the world already. But it's time for us to start visiting those beautiful, famous national parks here, so I'm really excited about that. So that's really what is on our bucket list for this year. So, a nice combination of work and also have relaxation and look at the beauty of nature, so that's what we're looking forward to.
Oh, that's wonderful. You can't beat it. Do you have a what's your short list of the ones you want to get to first?
Well, obviously, I think everybody. We'd love to see Yellowstone, and
Yellowstone, yep.
and there's so many gorgeous ones. And I think the first ones you're going to visit, I think, is Zion and Briar. So I think those are the first ones. And then we'll see. But it's the challenge always is to find the time to do it. I think that's that is time is critical in many aspects, but including in your own personal life for relaxation.
Sure. Well, I hope you get to as many as you can this year, and that each one provides some new and interesting adventures as you check them off your list.
Thank you, Erin. Looking forward to it.
Yes. All right, listeners, that wraps up this episode of Cell and Gene, the podcast. Thanks to Triumvira's Paul Lammers for being my guest and for all the amazing insight you shared with us today. So, Paul, thank you for your time. This was fantastic.
No, you're welcome, Erin, anytime. So have a great 24.
And thanks to you listeners for tuning in. Check out cellandgene.com for all the information you need to perform your cell and gene therapy role better. And we'll talk to you soon.